talking about the pigment system, students, so you know the various pigments here, chlorophylls and carotenoids, all of that. So the pigments, they are organized in two discrete photochemical light harvesting complex that's known as LHC within the photosystem I, and photosystem II. So these pigments, they help to make the photosynthesis more efficient by absorbing the different wavelengths of light. And the single chlorophyll A molecule forms the reaction center, and the reaction center is different in both the photosystems. Remember that. So you see the light in the form of photon is falling on the accessory pigments and the chlorophyll A, and all the light from the accessory pigments will also be transferred onto chlorophyll A, and thus excitation takes place. We'll come to that. So in photosystem 1, let's first talk about photosystem 1. So photosystem 1, the reaction center chlorophyll A has an absorbing peak of 700 nanometer and hence is called as P700. Whereas when we talk about photosystem 2, it has an absorption maximum at 680 nanometer and we call it as P680. So you see this is P680, that is the photosystem 2, and photosystem 1, that is P700. And that's a Z scheme that we're going to talk about. So this is the photosystem 2 here, and this is the photosystem 1 here. Now photosystem 1 or photosystem 1, which we are talking about, photosystem 1 or 2, the most important pigments of the system are chlorophyll A 670, chlorophyll A 683, chlorophyll A 695 and P700. So some physiologists also include carotenes and chlorophyll B in pigment system 1, and thus this system absorbs both wavelengths that are shorter and longer than the 680 nanometer. So we see here this light falling on the photosystem electron accepted. This is the reaction center, that's the chlorophyll, antenna pigments, and this is the whole of the photosystem. Now under photosystem 2, students, photosystem 2, the main pigment of this system, they are chlorophyll A 673, P 680, chlorophyll B and phycobilins, and this pigment system absorbs wavelength shorter than the 680 nanometer only. Now let us try to compare photosystems, that is photosystem 1 and photosystem 2, on the basis of location. So photosystem 1 lies on the outer surface of the thylakoids, whereas photosystem 2 lies on the inner surface of the thylakoids. On the basis of evolution of the molecular oxygen in this system, molecular oxygen is not evolved, whereas when we talk about photosystem 2, as a result of photolysis of water, molecular oxygen is evolved. On the basis of the reaction center, students, in photosystem 1, its reaction center is P700, whereas in the case of photosystem 2, the reaction center is 680. Formation of the NADPH. In the case of photosystem 1, it is formed in this reaction, whereas in photosystem 2, NADPH is not formed. On the basis of the cyclic and non-cyclic photophosphorylation, students, that's very important to know. In photosystem 1, it participates both in cyclic and non-cyclic photophosphorylation, whereas when we talk about photosystem 2, it participates just in non-cyclic photophosphorylation. On the basis of the source of electrons, students, in photosystem 1, PS1 receives electrons from photosystem 2, whereas when we talk about photosystem 2, it receives the electrons from photolytic dissociation of the water itself. When we talk about the involvement of the photolysis of water, in which photosystem involvement of photolysis is taking place. In photosystem 1 is not related with the photolysis of water, whereas photosystem 2 is definitely related with the photolysis of water. Now we land upon to another question, and the question is, electrons from excited chlorophyll molecule of the photosystem 2, they are accepted first by, is it cytochrome B, is it cytochrome F, is it quinone, or is it ferredoxin? What do you think is the answer? The answer here is, it's a C, quinone. Now talking about the pigment system, students, antenna or accessory pigments receive radiant energy and transfer it among them, and this transfer of energy is known as resonance transfer. Students, here in you see antenna or the accessory pigments, they receive the radiant energy and that is being transferred among themselves, and this transfer energy is known as resonance transfer. Then this antenna gets molecules that are excited and transfer their energy to the chlorophyll A, that's the reaction center. That's the molecule of the reaction center, and this is known as the inductive resonance. So all the accessory pigments will collect the energy and then send it to the reaction center, which is known as the inductive resonance. As you can see in here, the light has been connected, harvested, and then it's passed on to the reaction center. Finally, chlorophyll A molecule converts the light energy into electrical energy by bringing about electrical charge separation. Okay, so here you see photons, the reaction center, the pigment molecules, and here you see the primary acceptor.